Hi, everybody. Today we are going to talk about part 3 of cell signaling. So we are going to be talking about transduction, so the second phase of cell signaling. We're not really going to be talking about phase 3 very much. So before, we talked about reception, and we talked about how there was cytoplasmic reception. That one was relatively simple. And then there were gated ion channels. That was a membrane receptor. And we had two other membrane receptors, including the tyrosine kinase and the G protein. So today we are going to talk about what happens after that. So a few important things to keep in mind here. The same signal may produce different responses in different tissues. This is really a key point that when your body, when one of your cells releases a signal, it could attach to a few different types of cells, and those different types of cells can react differently. A perfect example of this is epinephrine. So epinephrine, for example, can bind to receptors on heart muscle cells where it stimulates contraction. Your heart is going to start beating faster and faster and faster and faster. But it can also bind to smooth muscle cells, which are the muscle cells that line your blood vessels and your digestive system. And there it is going to slow the muscle contraction. So how can that happen? How can we have two very different responses, the same signal? And the explanation here is it's the transduction phase. It's the things that happen in transduction. The things present are different in those cells, and so they produce different results. So these events, which are critical to a cell's response, can be either direct or indirect, and we'll talk about what that means. Direct transduction is a function of the receptor itself and occurs in the plasma membrane. Okay. Direct transduction is usually going to involve a lot of proteins, whereas indirect is not. Indirect transduction, which is more common, another molecule termed a second messenger is going to mediate further interactions between the receptor and the cell's responses. And we will look at examples of these so they make more sense. All right. In both cases, the signal initiates a cascade of events in which proteins can interact with other proteins until the final response is achieved. So a cascade happens when one thing activates a different thing, and that different thing, that protein, is then going to activate something else, and it's going to activate something else, and it's like dominoes. One thing activates another, which will activate another, which will activate another. Cascades are important because they can help with amplification. This can make a very weak signal very strong. And we'll also talk about amplification a little bit. So let's first talk about direct. Direct transduction usually involves a protein kinase cascade to amplify a response. So some keys here: catalyzes the phosphorylation of target proteins. That's going to be a really big deal. All right, so what does that look like? Here we have a tyrosine kinase receptor. It's activated, it's been phosphorylated, and we now have these activated proteins. So this is an example of direct transduction, right? We have a protein on protein activation, and that is considered direct transduction. Right, we'll be watching these videos tomorrow. So here we see a more general view, but also a more complete view. So up here, where my pointer is pointing, we have an activated receptor, and there has been a conformational change on the cytoplasmic side. This conformational change on the cytoplasmic side has activated a different protein. So one thing to keep in mind here, this blue protein and this part, these are two different proteins. Make sure you understand that. The cytoplasmic side of the receptor is going to touch this protein and activate it. So it has caused a conformational change here, and this activated protein is actually going to activate a different protein. So now we have a third protein. This is an inactive kinase. And once these two things interact, this inactive protein kinase is going to become an active protein kinase. So notice This is like dominoes falling, like we talked about before. So just to clear up any misconceptions, this protein here that I'm highlighting with the pointer, this did not become an active protein kinase. This protein activated a different protein kinase. So this is a protein kinase, and as we know, a kinase is going to do something very specific. Think about what that is. A protein kinase is going to phosphorylate another thing, in this case, another protein. So this active protein kinase is going to meet up with yet another, so this is our fourth protein already, another protein kinase using ATP, using ATP, the first kinase is going to phosphorylate the second kinase, activating it. That phosphate group right here, this thing is going to stay on this protein kinase 2, because that's what makes it active. And this then is going to use a phosphate from ATP to activate yet another protein kinase. This one stays active and it continues to, like an enzyme, it continues to activate a bunch of protein kinase 3s. So then this active protein kinase is going to activate another protein. And finally, we're at the end of the cascade. This one then can cause some sort of response, and that response can vary. It could start translation in the nucleus. It could open up in an ion channel or put some other carrier protein in the membrane to allow something to flow in or out. It can activate or deactivate other enzymes. There's a number of things that can happen. Okay, so think about what's going on there. This is fairly complicated. Make sure you understand that this is a cascade of one thing activating another, and that other thing activates yet another thing, a third thing, and will activate a fourth thing, activate a fifth thing. So we have this constant series of dominoes. Now, another big thing here is the idea of amplification. Amplification is an important idea because this receptor doesn't just activate one relay molecule, it can activate a bunch. Let's just say 10, even though it's probably a lot more than 10. So now we have 10 activated relay molecules. Each of them can activate 10 protein kinases. So now we have 10 times 10 or 100 activated kinases. Each one of these 100 activated kinases can then activate 10 protein kinase 2s. So now think about how many that is, and then they can activate 10 more, and they can each activate 10 more. So at the end of this cascade, we have amplified the signal exponentially. Activation is very, very important. Amplification is very, very important. Here we have an example. What type of receptor is this? So this is one of those tyrosine kinase receptors. And as we know, when it is phosphorylated and activated, it can activate a protein. And now that protein can activate a protein called RAS. RAS can activate a protein called a RAF. RAF can, once it's active, can activate a protein called MEK. MEK can activate a protein called MAPK. And MAPK can then, once it's activated, go in the nucleus and perform some sort of cellular response, which could be to start transcribing DNA. So this cascade here, and we know that at the end of this, because of amplification at the end, there's going to be a lot more than one MAPK, isn't there? There's going to be thousands or more activated MAPKs. Here we have another view of a cascade in amplification. Again, we have RAS activating a protein, activating another, and finally we have a response protein that's going to do something. So protein kinase cascades are useful because they amplify the signal at each step. Each newly activated protein kinase is an enzyme which can catalyze the phosphorylation of many target proteins. The information from a signal that originally arrived is communicated to the nucleus, if that is the intended target. And another important thing that sometimes people forget about, the multitude of steps provides specificity to the process. So as we have learned with epinephrine, when a signal binds, it might do different things depending on which cell it's in. This is probably not due to the receptor. This is probably due to the transduction phase. So if we have different proteins in these types of cells, then the protein cascades are going to be different, and we're going to end up with a different result, which is why your heart might beat faster, and the same signal might cause the muscles in your digestive system to move more slowly. Okay, now indirect signal transduction. So direct involves proteins, indirect is going to involve other chemicals that are not proteins. There are two types of second messengers. We're only this year going to talk about CAMP. We are not going to sadly talk about PIP2. Both of these things are second messengers, and that is what indirect signal transduction involves. It involves something called a second messenger. Second messengers are substances that are released into the cytoplasm after the signal is going to bind to the receptor. In contrast to the specificity of receptor binding, second messengers are going to affect many processes in the cell. So there's actually a number of things that a second messenger can do, whereas proteins are very specific. And that makes sense, right? Because they're enzymes, and we know that enzymes are specific. Second messengers are more general chemicals, and they are going to cause a few things to happen at once, usually. So they are less specific. All right, so here we have the example of a G protein receptor. So we already know how a G protein works. We've activated the G protein. It's going to an effector cell, and the effector cell is going to activate a second messenger. In this case, it's going to take ATP and turn it into something called CAMP. And usually what happens with a second messenger is often a second messenger is then going to activate proteins, and then we're going to end up with a phosphorylation cascade like we saw in the first part of this lecture, and that is what steps 5 and 6 are showing. All right, we're going to watch a video on this later. Like protein kinase cascades, second messengers still amplify, because we can make a lot of them. A single epinephrine molecule, for example, leads to the production of many molecules of CAMP, which can then activate many enzyme targets, because second messengers do a lot. Second messengers themselves do not have enzyme activity. They aren't proteins, they're not enzymes. They're going to act like cofactors, remember those, or sometimes allosteric regulators of a target enzyme. CAMP is a common and widely used second messenger. Adenylyl cyclase is the effector enzyme that is going to make CAMP from ATP. So ATP is the inactive second messenger, CAMP is the activated second messenger. Usually CAMP is made from a G protein. So a G protein receptor is usually going to be with production of CAMP. We'll watch this video tomorrow. CAMP has two major targets. Number one, CAMP can bind to an ion channel sometimes and open them. CAMP can also bind to a protein kinase, as I already mentioned, and activate it. All right. So here we have an example. So this is a G protein. We have a ligand. So in this case, it might be a neurotransmitter has activated our G protein. There's our activated G protein heading to our effector protein. Our effector protein is adenylyl cyclase. It's activated, and once adenylyl cyclase is activated, it's going to take ATP, turn it into CAMP. CAMP is our second messenger, and you can see that there's two arrows coming from CAMP, because CAMP can do many things. One of those things it can do is it can activate an enzyme, so it can start a protein kinase cascade, and at the same time, it can change membrane permeability by opening up that ion channel. So adenylyl cyclase is churning out a ton of CAMP. These CAMPs can activate enzymes, and they can change the permeability of various membrane proteins, opening up those ion channels. All right, so here we have a specific example of epinephrine. Epinephrine is going to be a ligand that induces what's called a fight or flight response. So let's say that you were, you're walking down the street and you see a lion. All right, so suddenly you panic. What are you going to do? You can fight or you can flight. Both of those things, when you come face to face with a lion, are probably very stupid. But you have, that's your natural response. You want to run or you want to fight back. Hopefully not against a lion. So in this case, epinephrine is produced, it reaches a receptor, and the receptor activates a G protein. The G protein is going to activate adenylyl cyclase. You see that here, follow my pointer. Adenylyl cyclase is going to activate CAMP. Remember, CAMP comes from ATP, so there's CAMP. And CAMP is going to do two things. One of the things that it can do is it can deactivate glycogen synthase. Now, this should actually make sense because glycogen is storage of energy. Well, if you are about to fight or flight, you need that energy. You don't want to store it. So inactivating the thing that makes glycogen is helpful. And the other thing that CAMP is going to do is it's going to activate a protein kinase. And that protein kinase, right, we already know how this is going to go. That protein kinase is going to activate another protein kinase, which is going to activate another protein kinase. And that is going to activate glycogen phosphorylase. What is that? Well, it's going to take glycogen. Remember, glycogen is stored carbs, and it's going to break glycogen down into glucose, giving us energy. And what can we do with that energy? We can use it to make ATP so we can fight back against this lion. So that is how a second messenger is going to work. You can see how it's related to the G protein, and you can also see how it's related to a protein kinase cascade. And in some cases, it didn't even use a protein kinase cascade. It went on and did something else. There are other second messengers. CAMP is only one. We do not have time to go over DAG and PIP, but I did want to show you a picture of how it works just so you can see another example. So in this case, we have a G protein, and the G protein hits an effector cell called phospholipase C, which is going to break off. It's going to hit this protein called PIP2. Well, it's actually a lipid. And it's going to produce two second messengers. There's DAG, which will stay in the membrane, and then there's IP3, which is a second messenger that's going to do a bunch of things in the cell. So there are other second messengers besides CAMP. All right, well, that is where I'm going to leave you for today. Direct and indirect transduction. Let me know if you have any questions.